My topic today is activity/block architecture, and I'm also going to discuss the cellulose like for human pathogens. First, there are 5 parts in my content: introduction, methods, results, discussion, and conclusions, future directions. And first, this is our background. In the context of global shortage of water resources, wastewater treatment is extremely important. And activity/process is the most widely used method in the wastewater treatment. And consists of flocks with strong absorption and decomposition capabilities. And research on flock formation is very critical. It's critical for wastewater treatment. But currently, there is still insufficient understanding for the flock formation. In order to explore it, we established an experiment model of activity/flock formation based on the cellulose as a substrate. In this experiment model, we hypothesize that activity/bacteria forms a biofilm around the cellulose particles. And in the experiment, we use a cotton ball and a toilet paper as substrate to represent the cellulose particles. Next part is methods. We prepare 6 bottles and pour an equal amount of sludge into each of the 6 bottles. And these 6 bottles are divided into 2 groups. Cotton ball groups and toilet paper groups, and we label them with C1, C2, just like that table. We smash the toilet paper and the cotton wool ball into small balls and place the 4 corresponding balls in each of the bottles. Then we put the rubber tube used for aeration into the bottle and plug the bottle with cotton wool. Next 3 steps are DNA extraction, sequencing, and Illumina sequence analysis. This 2 table shows all the samples we have taken during the 2 weeks. This is parallel samples, and those are blank samples. The parallel samples are divided into 3 groups. Which were taken on the start day of the experiment, and week 1 and week 2. And the sequencing step is divided into 2 parts: DNA quantification and amplification with PCR technology. This is our results. This table shows the relative abundances of different bacteria in the sludge. I selected 33 representative bacteria listed separately, and the remaining bacteria are classified in the category named Other. That's 35. We can say that the bacteria growth on the cotton ball surface is a little different from toilet paper surface, but we can say that they have the same growth patterns, variation patterns. In the initial stage of incubation, we can say that the number 5, 19, and 31, these 3 bacteria have the relative high relative abundances. And in the last stage of incubation time, the number 6 and 8 and 28 becomes relative high. And overall, the bacterial community composition on the cellulose surface changed from Cyprus Beryl and Delta Protobacteria to the Bacteroideae and Beta Protobacteria.
Okay.
And this is our second result. These two figures show the level of diversity of different between